and some more stuff here. And so we want to start with, she kind of described the whole picture. You're doing an experiment in your lab, but you need to communicate with us so that when you're finished with that experiment, the samples are collected correctly, you have enough material, certain buffers are used or not used, whatever, so that the best experiment can happen. When it comes onto when those samples come to us, there's certain things that can't be undone. And so we want to make sure that you guys understand that we communicate clearly. It's not, some of you guys have core facilities on campus where you just sign up for something and you go in and use a piece of equipment, or you drop samples by and you get a readout. It's a little bit more of an intimate relationship here for us to do proteomics well. And that's what we found that's been most successful, is the communication up front and on the back end. Because once we get those samples, it's done. So let's talk about what kind of proteomics we do, because that word is big. Proteomics means basically working with any sort of group of proteins. We do bottom-up proteomics. We like to digest our proteins into peptides. So even though there are tens of thousands of proteins in a sample, we like to make our job even more challenging by chopping those up into peptides. And we do that because proteins are really sticky, and peptides can be made into solutions, which then solutions can be transferred much easier, and then we can be separated. So the peptides that we want to do, that we would like, are peptides that have about 7 to 20 amino acids in length, and that's kind of a variable number here. And so we will use some sort of proteolytic enzyme. Usually we use trypsin. We don't have to, but trypsin is by far the most efficient and specific, and so everybody does. And little known fact, trypsin cleaves at the arginines and the lysines, and that's about 10% of the amino acids. So you get this nice size that works well with the mass spectrometer. That size is based upon what will fly through the mass spectrometer. So think of that proteolytic enzyme as a giant pair of scissors chopping up your protein at very specific places to form not just peptides, but triptych peptides. So they have a lysine or an arginine over there on the C terminal. And then we take those triptych peptides, we separate them by liquid chromatography, which is no trivial thing, okay? Everybody says, oh, I got this great big cool new detector. Like, yeah, but can you run the LC on the front end? Because that makes all the difference in the experiment. And then, of course, you have a mass spectrometer that does some measuring there. So how are we able to separate those peptides? So I want to speak a second about the liquid chromatographic system here. So the goal of liquid chromatography is to separate those peptides into distinct groups. And it separates them by its hydrophobicity. So more hydrophilic means they're smaller. The peptides are smaller and they have a higher charge. Peptides can be +2 charge, they can be +3 charge. And so the higher the charge, or the smaller they are, the earlier they elute out of the system. And then the hydrophobic ones are larger and they're less charged, but they elute late. So what the goal is of this whole liquid chromatographic system is to crowd those peptides into concentrated groups that can be analyzed individually. So we're not actually putting them in bins. It's one giant continuous thing, but we want to line them up. It's like if I had everybody leave this room at the same time and everybody's going, but I want you guys to filter in. So all the first years go first and all the second years and the faculty go at the end. And, we separate everybody in groups like that. And so if you think about it in your head, we have peptides that are all jumbled together. They all have different sequences. And then when we do our chromatography, we're still going to have peptides, but now they're going to be marching out in a very specific order here. So a couple of things will happen. One, you're going to be able to analyze a certain peptide only at one particular time because it's all coming out at one particular time. And when they're all crowded together, now the signal for that peptide is much bigger than just a single molecule event, which may not be readable, okay? So how are the peptides separated? Well, let's get down into the minutia here. And I think I brought my prop today. Don't drop it. So this line here on the screen is about a 15 centimeter line. At least it was on my computer screen when I made it. It's a little bit bigger here. But that is about the size of the column that we do all of our separation in. And I'm passing it around. It's a piece of capillary that is 75 micron in inner diameter, 360 micron outer diameter, that's filled with a bunch of really, really small beads or resin. Then this is a micrograph of one of those. And hanging off of this micrograph is going to be some chemistry that is going to retain those peptides as they come by. So the LC system is made up, really, most of that stack in that previous picture is a lot of really expensive pumps that deliver high pressure, very low volume, okay? And to make very consistent flow rates. That's the whole goal. The peptides are separated with a column. And again, the particle size is about 2 microns in size, is the size of this particle. So you're taking this really small column and you're packing it full of really small particles. And now you're trying to push liquid through it. So it requires really high pressure to do that. And that's why you have to have really expensive pumps to be able to do that whole thing. And so then hanging off of there, you've got this long carbon chain. Usually it's a C18, some sort of derivative of a C18 chain that will give that hydrophilic, hydrophobic, they call it a helic chromatography. That basically the whole idea is we want to retain the peptides just a little bit, but not very much, because what you don't want to do in chromatography is have too much retention, because then you can't get the stuff back off again. And it makes a really long retention time, and then the stuff starts to spread out. So you want to basically just have just a little bit of a delay. And so a lot of technology has gone into the size of these particles, how these chemicals are attached to it, and all that kind of stuff. So how do we detect and identify the peptides? Well, to keep those peptides, we're thinking downstream, we want those peptides to be charged, because the mass spectrometer is really great at charged ions. It doesn't do so well if it's not charged, because it can't move things if it doesn't have a charge. So we want to keep those peptides positively charged. We know that the C terminal is going to have a lysine or an arginine hanging off of it. And that is a basic residue, right? It has an R group. The lysine has an R group that has a nitrogen on there. And so in an acidic environment, it's going to accept a proton and it's going to become positively charged, okay? The other end of the peptide has the N-terminal end. So there's at least 2 nitrogens on every tryptic peptide that can accept a proton. And that's why we say most peptides are a +2 charge. Now, some of you are going to argue and say, wait a minute, there's some peptides that are +1. You're right. And we throw all those out. Because we do. What you can do with the mass spectrometer is you can tell it, hey, only look at the +2 peptides or the +3. Don't look at any +1 charged. We call these precursors because we don't know if they're a peptide or not. But almost, let's put it this way, 95% of all +1 charged precursors are going to be contaminants. We're going to lose about 5% of our peptides if we just filter by getting rid of all the +1s. So some people will keep all the +1s. That's fine. We don't. We say +2, +3, +4. We usually go from +2 to +7. So you say, well, where do those other pluses come from? Don't forget the other basic residue. Come on, who wins a golden star? Histidine. Yes, that's right. Histidine is the other basic residue that could be in the middle there and could also has a nitrogen that can accept a charge in it. And so to get them positively charged, we put them in an acidic environment. Those peptides have a specific mass, and the mass spectrometer measures mass to charge. So the first thing that we want to do is get just a scan, what we call the MS1 signal. But the mass spectrometer can also fragment peptides. So it can scan and kind of take a snapshot and say, what are all the mass to charges ratios in the intensities of all of the precursors that are coming out at this moment in time? But then a mass spectrometer can also hone in and take a collection of those and put them in a separate chamber and hit them with energy. And that's what we call the MS2 signal. Okay, we're going to talk more about that. So here's the idea, right? Chromatography got all those green peptides to come out at the same time, and those green peptides all have a positive charge. They're charged. Then we can just focus in on the green peptides versus the yellow ones and the blue ones. We can hit it with some energy, and we get a bunch of green fragments. Those green fragments, all of those are going to somehow point to the identity, the actual sequence of that green peptide. And we need to know that piece of information because the mass to charge information is important, but it's not going to tell us the identity, the actual identity of the peptide. It's the MS2 signal that's going to do that and these fragments. So what does the initial data look like? Okay, and later, I think tomorrow, we're going to talk about some of the different files that you get after a bottom-up global proteomic experiment from us. And the files can be overwhelming. There's a lot of different files, and what do I do with this file, and what information do I get from that file? This is what we would call a chromatogram from the raw file. This is the data as it's being collected right off of the instrument. And so we have time down at the bottom, we have relative abundance over there on the side. And so this is what we call the gradient. As we increase the organic nature of the solvent that's flowing through our column, peptides are going to come off based upon their hydrophobicity. So the really hydrophilic ones are going to be coming off here. The really hydrophobic ones are going to be coming off here. And we could have 100,000 peptides or precursors all in a single 30-minute run. That's a lot of information. And you might be thinking back to your chromatography class. If you took one, you're like, that is not baseline resolved chromatography. You're right. Stuff is crowded out. It's coming off. We want to crowd these guys together and we want them to come off, and they're coming off. But you got to remember, we have a mass spectrometer that can tell the difference between these guys. So we don't need baseline resolution. So the goal is to have, our goal in chromatography on the front end is to have these peptides spread out over the time window by the gradient so that the detector can have the best chance to detect as many things as it can, because it's really a time resource thing with the detector. It can only look at so many things at one time. So if we're interested in, say, that guy right there, then all we're seeing right now is what we call the total ion current. That's where this intensity comes from. It's just saying this is the total ion current that came out at that specific time. If we were to take this guy out, almost like a third dimension, you could slide that out and you could tilt it, we would get this signal right here. This is the MS1 signal. That scan at that particular time looks like this. So each signal or group of signals represents an ion, might be a peptide or precursor. And I used to go into the isotope envelope so that to show you how you could calculate whether it's a +2 or a +3, and I think that's really cool, but we're not gonna do that today. But if you want to know about it later, Zoom me and I'll be happy to show you. But let's say like that little guy right there, and it could be a peptide, right? Or this guy could be a peptide, or this guy could be a peptide. Which is the guy that you may be most interested in? The big one, that's right. And that's how they make mass spectrometers. And for a long time, data-dependent acquisition would take a scan like this, and it would say, we're going to start with the most important, the most intense peptide here, and we're going to look at that one first. We're going to isolate those ions, fragment them, look at the MS2 signal. Then we're going to go to the next one, and the next one, and the next one, and the next one. And it's going to do that for a couple of seconds before it says, okay, I've got more stuff coming out. I got to take another scan, and I'm going to do that whole process again. So yes, that could be the most important one. But I don't want to burst your bubble, but that's probably albumin right there, or hemoglobin, or actin. Sorry. Where is your peptide that you're interested in? Probably there is what is probably the guy that you're interested in there. And so for data-dependent acquisition, this method of identifying which ones are the important ones based upon size is not really practical, because you will lose, a lot of times you will never even, the detector won't even get a chance to get to this one because there's so many other things it's seeing at the same time. Okay. This is lead up because there's a new guy on the block called data independent acquisition that would basically do a different method. And what it would do is it's going to take and put things in bins. Instead of going from isolating each one individually like this, it's going to isolate all of the peptides in a certain bin, say from 400 to 420, 420 to 440. And it's going to look at all the peptides and it's going to fragment them all at the same time. But I don't want to get ahead of myself. So how do we interpret the MS2 spectra? So there's been some collision gas. So the whole mass spectrometer is under vacuum. We're going to get to that in just a second. But we would isolate some ions, route them into a certain place, and then we'd hit it with some high energy collision gas to break the peptide into fragments. That break usually occurs at very specific points on the peptide backbone. Very interesting stuff. Depending upon the gas that you use and the level of energy, you can get those breaks to occur at different places. And that's really can provide a lot of really useful information. The loss of mass, then, if you break it at the right place, will actually equal an amino acid that's falling off. So each fragment, mass to charge ratio, is going to be one amino acid in mass different from the next guy. So this is my representation of my peptide. You got your C terminal here, you got your N terminal down there. Each one of those R groups represents a different side chain hanging off of there, which is the identity, whether it's alanine or valine or leucine or whatever. And then these little dotted lines here are the breakpoints. And the Y ion, B ion breakpoint is the most common for the type of chromatography or the type of mass spectrometers that we use. But you can also get these breaks along the A ions, and there's Z ions, and there's all kinds of other breakpoints here. So this is what an MS2 spectra looks like. So just imagine back, we took MS1 and the mass spectrometer was actually able to isolate just one particular set of ions, put them in another part of the mass spectrometer, hit it with some collision gas, and it broke it apart into fragments. And so now what you're seeing is each one of these guys here is a fragment from the same peptide. And so, if Rick were here, I'd make fun of him because he's been doing proteomics since it was, born. When he was in graduate school, he would take these MS2s and sit down with a calculator and his pencil, and he would figure out what the structure was by hand. We have these things called computers now. That's what I tell him. That does all this for us. But the mass difference there between these different ions will tell us the signal. So the Y1, the Y2, the Y3 are in these things. So just think about these signals here are no longer green peptides. They are fragments of the green peptide. And if we put all those guys back together, it would be like this. If I took this guy right here, if I took that peptide and I hit it with energy, when it fragments, it's going to fragment in a way where it has a different, you can see the amino acids are falling off. So we're losing the glycine or we're losing the proline or whatever here. And those are all equating to a different signal here. And so if we just subtract the difference here, we get a certain mass that then equates to a different amino acid. It's beautiful. So now we can have identity. This is like, you look at this MS2 spectra and it's like a fingerprint, right? Except that our assumptions are based upon that this is just primary structure and that this is what every peptide looks like. But we all know the ugly truth, don't we? There are things hanging off of there. Modifications, and that can make this harder to interpret. And we also have other things that we're all ignoring. What's all this stuff down here called, this stuff in the grass here? Call that noise. Yeah, so there could be a noise signal in a place that then confuses the program that is trying to make the determination. And so we still need humans to do this. Okay. So let's keep going. What type of tryptic peptides do we make and detect? So I just chose one guy. I chose from the human FASA file of all the proteins, this human insulin-like growth factor 2, and then this I went to all the painstaking work of coloring what the different possible tryptic peptides would be like. So this is what's great thing about, since you know that trypsin is going to cleave at the C terminal of all the lysines and the arginines, you can do what's called an in silico digestion. You can figure out what peptides you would probably find before you actually do the experiment. And there's lots of tools online that will do that. And so that's what the red and the blue are. But the typical digestion here for this guy, we have 605 amino acids in this protein. Some of these after digestion, we may have, you'll notice that there's a couple of places where there's an arginine right next to a lysine. We'd get a single amino acid. We're never going to see that. And it doesn't make any sense. Everything's got a lysine. But we also have some peptides that are 58 amino acids in length. That may not fly through our detector. They may not get off our column. That may be too sticky. It's too hydrophobic. It's got a great big stretch here of a lot of things. That's probably that guy or maybe this red guy here. But they'll end up with 70 different possible peptides. So the question that people always have, they're always a little bit amazed is, how many peptides, what is my coverage? How many peptides did I actually see for that protein? Well, first of all, not all those peptides are going to be ionized and detectable. Some are going to be too small. Some are going to be too big. Some of them just don't fly well through the mass spectrometer. There's going to be gaps in the sequence and the coverage, but it only takes 2 unique peptides to have a score above the predefined threshold to identify a protein. Now, in this day and age, I see people that go down to 1. They say, I only need to see 1 peptide. Yeah, Ryan.
What sets, earlier you mentioned the lower limit is about 7. What sets that lower limit of 7 amino acids for a peptide? Is that a physical property? Is that sequence identification?
Yes, I mean, sometimes we can see peptides that are 5 amino acids, but here's kind of an interesting thing, Ryan. If I were to take any 5 amino acids here in a sequence, and I just were to open up my FASTA file and I were just to say find, I would find dozens of examples of 5. Once I get up to about 7, you can start to get to unique. And that's the thing. So it's not that we can't see a 5 amino acid peptide. It's just, it won't help us with identification. So a little guy like that one would be too small. So what does the actual coverage look like for this protein? And this may shock you that for this is a real sample here. What we see in gold here, that's the actual number of peptides that we actually saw for this human insulin-like growth factor. And if we zone in on just this one peptide here that starts with the methionine here, that guy right there, some of the software out there will show you an output that looks like this. It gives you spectral level information. This is the MS2, so this is DDA experiment. It actually isolated that peptide, that M over Z. It fragmented it, and what it's telling you is it's saying all of these blue signals here line up with the Y ion series. It actually has a table like that in silico digestion that I talked about. We can already predetermine what the masses that we would be for the Y ions and the B ions, and sometimes we have water loss and ammonia loss, and there's all kinds of chemistry going on in the gas phase here. But you'll also notice that there's a bunch of noise down here as well. So some of the nuances of being able, when you're trying to hang your hat on a certain spectra, this is where you want to, like, some spectra, all spectra are not created equally, right? So I would be much more comfortable with some of these Y ions out here because they're in a region of the spectra that has a lot less noise than maybe some of these B ions here that are much smaller and in a really noisy spot. Does that make sense? And so this is the kind of level that most of the biologists go beep, because you're like, whoa, we are so far from my rat model study now. We're looking at little squiggly lines on a thing. But this is, you got to think about, like, you're making biological decisions on what proteins to go chase based upon this level of information. So at some point, you gotta either do it yourself or know a guy that can come in and be able to tell you Yeah, that's valid. That's real. Okay. And that's part of the collaboration that we have here. So how do we know what proteins we're looking for? Because some of you guys are still thinking that we do magic, that we from nothing create all of the peptide sequences that are in there. No, we have a library and we search against that library. Okay, so because of our folks over in genetics, we know that we can do sequencing. And for the human, for instance, proteome, we can go to this great website called Uniprot. If you haven't been there before, you should go spend some time there. There's lots of great information. And it will tell you, for instance, the protein count, which I always tell people that this is the recipe. This is how you make a human. You just have to throw together this number of proteins and voila, out pops a human, right? But it tells you two interesting pieces of information. One is it tells you a gene count that's only about 20,000. And then it tells you a protein count, which is 80,000. And because we know those genes don't, the whole things kind of get in different places and all of that. And so you have isoforms and all of that, and all of that's included. And so it's really interesting because when people will talk to me and they'll say, I'm looking for a specific protein, and I'll say, okay, that's great. Is that protein part of a group? Because sometimes if it's part of a group, then it's really hard to tell you that specific protein. We can tell you that group because there are actually no unique proteins that point back, peptides that point back to that one protein. And so that's some information that we have. And here's another thing, and this may be a shocker, but people will say, well, I was looking for this protein and we didn't find it. Why? And I'll always ask this question. I'll say, is that protein in the library? Because sometimes you're like, well, we did this modification. We had this M cherry whatever into there, and I'm like, well, if we don't put that in the library, then we're not going to see it. And we can easily do that. It's really easy to add that kind of stuff in. So these libraries are super important. And so this is what an entry looks like for, again, there's our insulin-like growth factor protein there with its sequence, and that's what we have. And our assumption is that these proteins are known and finite, and this is an area of proteomics that is on the cusp. People say, you know what, that's not true. I want to create my own libraries. And some of you guys talked about these other omic experiments where you can say, I've got transcriptomic information that says what was that I want to be able to use. And so that's a conversation for Sam down the road. But for our mainstay, for the first run, if you send us a sample and you tell us they're human samples, we're going to go to Uniprot and we're going to get one of these libraries and we're going to search against that. Okay.
Is it based on like splice variants annotated in the genome, or
when you say they, what's
the library that they Uniprot? Is it based on different splice variants for each gene?
To get to the 80,000 versus the one gene per.
Yeah.
Yeah, I think so. I don't know. We're getting into a real fuzzy spot in my knowledge. This is Karen. Is it splice variants?
I think that's part of it.
Yes, ma'am.
Just want to know, like, even for bacteria where it's much simpler, the strain that you're using is so important because some of them are better annotated than the others. We actually discovered that there was, for the strain that we were using, the annotation was wrong for one of the genes because you guys by accident, well, the facility, but used a different genome from what we asked. And that was very lucky because then we discovered the whole piece of protein that was missing in the other annotation. So yeah, it does matter a lot which library you're using because they're So if you can broaden up your search, especially for species where you don't have too many options, or maybe to include some similar ones too, like they're like homologs.
Yeah.
For you.
Yeah, great comment there. If you guys don't mind, when you guys are making comments, can you make sure to turn your mic on so that the people on Zoom can be able to hear that too? So what have we learned so far? So we've learned a lot of things, but genomics has provided libraries of proteins. And this is a really good point that you want to be the driver of your project. And so looking into the library and seeing what that looks like for some of you, if you're new to proteomics, you may never have looked at the proteomic library and know that there are different ones. And NCBI has, there's lots of different things that you might want to look into there. Those proteins can be cut up into peptides, and peptides and the fragments can be identified by mass spectrometry. So that is essentially in a graphical sense, we have biological information. You guys are looking at phenotype changes because you're adding a drug, you have knocked out a gene, something. You send us those samples, we are taking the proteins out, we're doing a tryptic digestion into peptides. That hands it over into our data collection. We then use our fancy LC-MS systems to collect data, usually at the MS1, MS2 level. I'm going to talk about the MS3 level here in a few minutes. We do a database search. That's where these libraries come in. This is a really important point. How do we search the database? Do we look for modifications? What modifications do we look for? All those sorts of things. We come up with a list of proteins and peptides that we found based upon and on your samples and put those into groups. Each one of these signals here represent precursors or peptides. Maybe signals down here give us the fingerprint to identify the peptides. And then we end up with this list of proteins and peptides identified and their intensity. And then we start another experiment, right? We've already done 2 to this point. We start another experiment with that digital biological information of doing some statistics and looking at groups and doing differential analysis. Hopefully to be able to then provide some information back to you that you can look at and you go, oh, this makes sense. And this right here makes absolutely no sense whatsoever. All right, how am I doing? I know I've got 3 minutes to cover about 14 slides, so let's go. So here's the shock and awe thing, is that nano liquid chromatography is not for the faint at heart, okay? It's hard. And the reason is because the flow rate is, even though the pressure is really high, because the volumes are so small, the flow rate's really low. We flow at about 300 nanoliters per minute. That means that we will get 1 drop every 2 hours and 45 minutes. So unfortunately, what happens is that we have these really great pressure sensors that tell us that we have a leak, but we can't see it because there's no liquid coming out anywhere because it's evaporating before it actually collects enough to be able to see it. And so some plumbing is required here. So this is just a picture here showing you, I wanted to take a stock image from the website that shows the nice shiny instrument and show you what it actually looks like when you're using it. All the covers are off and there's tools everywhere and everything. And it's just probably your lab is similar with whatever you're dealing with. So the plumbing is required. And then what we want to do is sample prep finishes with peptides. We resuspend those peptides in a 0.1% formic acid solution. That's an acidic solution to make those peptides charged. And then we use a PCR plate here and put it, a couple tens to 100 microliters of that in there, and we put it in our autosampler, and then that gets injected into our system, gets separated through our LC, and then gets detected by our mass spectrometer. Okay, so the whole interface between the LC and the mass spectrometer is this weird 1 centimeter gap. And it just blows me away that this is where we are in 2026. So it's really hard to see, but this little line here represents the column that we passed around here. And I don't know where it ended up right here. So that column is where the separation occurs. And it's basically sitting right here. And there's a sharpened end to this that's essentially just pointing into the mass spectrometer. We have vacuum over here, and we're spraying out a garden hose over here effectively. And we are somehow in that 1 centimeter stuff happens, and what we want to talk about next. It's literally just a little gap. We just point this guy and spray. There's a spray here that nobody sees. This little camera is just to see if there's a drip on here. If you don't see anything, then everything's good. If you see a drip on there, things are bad. Yes, sir.
Sorry to ask another question. How does the pump maintain such small dead volumes? Because I guess I come from like protein purification field where our pumps have, we want to have small dead volumes, but it's like, 10 orders of magnitude larger than the volumes that you're
That's
trying.
like a Bradley question here, how do we keep such small dead volumes, Bradley?
So a lot of that comes down to the fittings and just minimizing tubing lengths. You use as small as possible inner diameter tubings. Viper fittings are really good because they're basically zero dead volume fittings that sit the flush in the bottom of um, I guess the other side of the fitting. So, yeah.
Yeah. And that's why when I say anybody can buy a detector, but it's getting all the right things and having that know-how and knowing how things get, because I hate to say it, these guys, they'll come and they'll install a new instrument and they'll have all of this stuff they'll hook up. And we wait till they finish with their calibration and then we throw a lot of that stuff away and we put our Viper fittings on there and we do, because their column that they have is good, but it's not as good as this other column that we like better, for instance, and we like this, whatever. So that's the way it goes. Okay, so just give you some idea. The tip of that column is running at 300 nanoliters per minute. There's about 3,000 PSI or more. And then we have the inlet to the mass spectrometer is under vacuum, just right there. And it's just pointed right at it. So let's talk about that just a little bit more. The electrospray ionization here, somebody won a Nobel Prize for this. I should probably know who that was. But essentially what happens is that as the liquid droplets come off and you've got all of these peptides that are ionized on here, it's like they don't like each other because they all have the same charge, right? Because they're all in an acidic environment. So they're all trying to stay as far away from each other as possible. The problem is that there is flow gas that is drying out these solvent droplets as they're being compelled to go into the mass spectrometer. Not just because they're being sprayed in that direction, but because we have a power supply that provides about 2,200 volts to compel them into the mass spectrometer. So as that solvent evaporates and those charges start to get closer and closer, they eventually get to the place where they just literally leap off and they form this Coulombic explosion. And you end up with these peptides in the gas phase, ions in the gas phase. I mean, it's just a beautiful beautiful system here, and this is what we do. And then those ions go in, and we have and it's all occurring in that tiny little gap right there. So I know most of you guys probably this is the part you dread, is where we start talking about the innards of the mass spectrometer. But we're gonna treat it like a black box, and then I'm gonna show you one little graphic here, and then we're gonna move on. But essentially, we have to have an ionization source, and that's what we just talked about, that electrospray ionization. And then you got to have a mass analyzer, and that's a vacuum chamber that somehow separates molecules that differ in mass. And we have different mass separators. We've got quadrupoles, we've got orbit traps, we've got ion traps, we've got these things called TOFs, time of flight. And so these are all different variations of mass separators that are able to do different things, separating and analyzing those. And then you have a detector, where you have a detector that converts those ions into some sort of electrical signal that can be measured. And you might think about it like, kind of like if you're shooting BBs at a target and every time you hear that ping, you hit one, right? Well, it's kind of like that. They have a detector that every time those ions hit there, it causes a little electrical charge or something, a cascade. The really fancy ones now, it's all optical. So now it creates a photon that then gets hit, that's pointed at a detector, and then it's all light energy. And then you got to have some fancy data system, because when you think about it, you've got ions that are coming in, ions that are being routed through different analyzers. It's keeping track of which ions it is, what the mass of the charge is, whether these are fragment ions, whether these are precursors, and all of that. And so we live in a beautiful age right now where computers are fast, the instrumentation's really fast, the technology's really good, and we were able to do, just compared to 5 years ago, so many things that we just never thought we could do. So I want to see if I can get over to this other. No, for a second here, just to show you this. So I think this slide is really cool. Okay, so this is not all mass spectrometers are built the same. This one is what we call a tribrid because it has 3 mass separators. It's got the quadrupole here. It's got the orbit trap, which essentially is put bottom up proteomics on the map. And then it's got an ion trap over here. And so here's the column. This is the LC system. And these little dots, these little spheres here represent the peptides that are coming in. And so what'll happen is those peptides come in. First of all, they always put a turn here. It causes the neutrals to go straight, and all the things that are charged will go around that corner. We're going to do that again. So you lose the neutrals. You take the things that have charge in here to the quadrupole, and the quadrupole separates and says, I only want to look at ions from 400 to 1200 m/Z. And it puts it into this ion routing multipole, and the ions literally stop, get turned around, go through this C trap, and get popped up into this orbit trap. Now, this orbit trap it looks just like that. It's a weird shaped spindle, and the ions precess back and forth across there. And as they precess, they have a frequency that then matches the mass to charge ratio. And so if you remember from NMR and all of that, anytime you have something that has a frequency associated with it, you can get much higher resolution. And that's a high resolution event there. So that's your MS1 getting high resolution information. That information now says, hey, I've got this scan, and I've got these ions of these mass to charge. And so I want to look at one of those. And so remember, the ions are coming in continuously. So the very next set, it separates out, this quadrupole separates out only the yellow guys. And once they're routed here, it can then put them over into this chamber where it gets hit with a collision energy, causing fragmentation. And then this ion trap would then measure the fragments. And all at the same time, you might have missed it, it's letting in the blue guys, and then it's going to let in the yellow, the green guys, and all of that kind of stuff. And so all of this is going on simultaneously. And so the name of the game is speed, trying to do all of these things very fast, letting ion packets in. And so our chromatography, our job is to try to get the peak width for each peptide to be about 15 to 20 seconds in width. That scan and then doing a bunch of MS2, every time we do an MS1, we do it about every 3 seconds. So let's say it's a 21-second wide peak width. We have 7 shots to be able to get that ion, identify it, to collect it, and to be able to identify it. And so that's the goal, is you got to know your chromatography, to know your peak width, to be able to know the speed of your instrument, and be able to couple all that together to be able to get all the information that you need.
Can I ask a question?
Yeah, please.
So the yellow guys and the blue guys,
Yeah,
are these just a specific type of peptides?
that's exactly right. That's right.
But how do you, if you have like 100,000 different species from MS1, how do you pick one of them and break into MS2?
Based upon the MS1 takes a snapshot, and it will go from the highest, most abundant precursor that it sees, and it'll isolate that one, and then it'll go to the next one, and it'll go to the next one, and that's what this quadrupole is doing. Its job is to be able to say, I'm going to take just the ions that fall into this massive charge window, and I'm going to route that.
So it does 100,000 of them separately. And is that
DDA does not, but DIA does. I don't know. I have to be honest with you. We've got an Astral, which is the highest frequency instrument that we have, and I'm not sure we've ever done a DDA run on it to see how many precursors. But with DIA, we can get above 100,000 precursors in a 30-minute run. Shocking.
Yeah.
I mean, it just in there, what it's doing is it's doing it by 2 Dalton or Thomson windows. And it's just looking at all the peptides that are coming out at that time, and it's analyzing them all at the same time. And so there may be a dozen all being analyzed at the same time.
And does it matter if some peptides are more abundant than the others?
It does. In DIA, it does not matter because it's just going to look at each bin individually. So if you have something that's really coming out that has a lot of intensity and you're doing a DDA run, the mass spectrometer is going to be overwhelmingly distracted by that. It's a bad day to do this, but if I ask you guys to look outside and count how many stars that you can see, you probably say, well, if the clouds weren't there, we could only see one. And that's what it's like when albumin's coming out during a DDA run, or hemoglobin, or
Keratin?
or keratin, something like that, something that's really abundant. But with DIA, it's looking at it's only looking at signal in this in this box, and so there could be something coming out over here, but it's just ignoring it. And so then it's looking at all the signal that's coming out in this box, all the signals that are coming out.
And by box, I mean like 2 kilodalton.
That's right, 2 kilodalton.
steps.
That's right. That's right. Yeah, right.
Sorry to
Thank
add
you.
another one. Is where in DIA is singly charged peptides thrown out? Or are they thrown?
That's a great question. You know what? Save that until after lunch when Rick comes. Any technical questions, please ask Rick. Because you guys know I've only been here about 5 years, and everything I've learned comes from that man. So it'd be easier to just get it from the horse's mouth. But yeah, that's a great question. Back here. Let's finish this up. But yeah, really good questions here. So just to broaden it again, I'm again an educator at heart, so covering information multiple times is not a bad thing. So if we were to go through this whole thing again, we can take, and we still do, we can take your gels and we can do a digestion straight out of a gel. We can also use, these are called fast filters. It's just a 30,000 Dalton cutoff filter, and that's really good for your pull downs, okay? And then our mainstay, and this is a separatory funnel, but it literally looks like that. We have a chloroform methanol extraction digestion protocol that we use. It's really good for especially things like brain matter, because brain matter has a lot of myelin in it, and that stuff is really sticky, and it tends to gum things up. And other things, this is really good because that chloroform layer can pull some stuff out. So we can take lots of different samples, and our sample prep people basically do the same thing. They get the protein out, they do a quant on it, they add trypsin, they get peptides, and you can say, well, wait a minute, what kind of samples you're talking about? I'm talking about clinical fluids, I'm talking about tissues, I'm talking about cell culture. People send us rice samples, milk samples. Every biological fluid you could possibly imagine has been measured, and they all have peptides in them eventually. And then that goes through our LC system, that goes into our mass spectrometer, that goes through the ESI, blah, 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 MS1, MS2. So you can see we have this, this is what we call the workflow. The idea is, or the job is can we get your samples with your criteria, your challenges up into that first part so that we can get us data at the back end that will then help you with your experiment there on the front end. Okay. You guys are going to ask about post-translational modifications. We can do some here, but almost all post-translational modifications, you've got a couple of challenges, and Rick's going to talk specifically about phospho, I think tomorrow. The challenges usually are they're not stoichiometric, so you have low abundance. Only some things are modified, not all are modified. And then we need to do some sort of enrichment. But we have successfully done phosphorylation and ubiquitination here. And we've also done some of these other guys. But I will tell you what we'll mostly tell you is if you'll do the enrichment on your side, then we'll be happy to analyze this, do the data collection on our side. But we don't want to be in the business. And here's the ugly word. Those enrichments require optimization. That's going to be specific for your samples and whatever kit that you're using. And so we'd rather you guys figure that out because there's too many variables for us to do all of that. So discovery quantitative proteomics, that's another way of saying we say discovery proteomics. That basically means we don't really know what we're looking for. We've made some sort of change upstream in the genetics, or we've added chemicals or something like that, and now we're wanting to see what proteins change. And so these experiments are designed to quantify and identify the greatest number of peptides or proteins in a biological sample. And we're looking for candidates. We want to sort these things out to be able to say which guys had the greatest fold change between my groups, which ones match my biology, which ones did I not expect, how do I, and all of that. But we can always increase the depth. People will say, well, I didn't see the protein I wanted. We only saw 5,000 proteins, and my transcription factor actually is protein number 6,500. And you think about depth in that we're going to see the most abundant protein first, then the next most abundant, and then the next most abundant. And so really it's about abundance, sort of. And so you can always increase that by fractionation, doing an offline fractionation where you take your sample and you have maybe 100 micrograms of just one sample there, and then you put it through a fractionator. So now each one of these samples has 10 micrograms of selected peptides. So you have the more hydrophilic ones here and the more hydrophobic ones there, for whatever. And then also more instrument time. The incredulous question of, can you really look at 100,000 precursors in a 30-minute run? Nah, you could do it better if it was over 2 hours, couldn't you? And so what we can do is we could set up the chromatography in the instrument to collect for a longer period of time. And the problem is there's the law of diminishing returns. As you start to do more fractionation, as you start to do more instrument time, you'll see an increase. But after a while, you've doubled your analysis time, doubled your money, and you've seen a 20% increase in your return. A couple other things I want to point out: the dynamic range challenge. Just looking at plasma proteome, the difference between the most abundant and the least abundant protein in there, it's 10 orders of magnitude different. And so that's the real challenge. And so when you guys come with your cell cultures and we told you to wash it a certain number of times and you don't, and we look at them and they have like a pink color to them, and we're like, we are going to see FBS like till the cows come home, because that is such a huge thing. And so that's the take home message is that if we could get rid of some of those really highly abundant things like that, then we can see some of the lower things. So for plasma, serum, and that, they make kits that will allow us to selectively remove the top 14 most abundant proteins, and that helps. The problem is that is species specific. It's designed for human, which means it works okay for mice and sort of for rat, but the farther you get off, then the worse it gets. I like to say this from the analysis point of view. The good experiments have simple or few comparisons. Some of you guys, you're getting into proteomics and you got this thing. It's like you're going to go buy your first new car, right? You are going to get every single option put on there. You're going to, you're just to trick this thing out. Proteomics, that's not necessarily the best idea. You just want to ask the most obvious question first. And you want to make sure you have enough statistical power. People always say, well, when I do my fill in the blank omic experiment, I only have to submit 3 replicates. Why do I have to do 5 for you? Well, because we have protein has lower between group changes, right? The changes are a little bit more subtle. We cannot amplify our signal like we can in some other omic experiments. We have contaminants. We have missing values. And we have greater in-group variability depending upon your sample type. So if you're doing cell culture, you might be able to get away with 3. If you're doing an animal study with tissue, I would say 5 is definitely the minimum. If you're doing clinical samples, then as many as you can get, right? Because the variability goes up. And I can guarantee you that you will figure out what you should have done with each proteomic experiment. That's what happens. So you're not going to, so going into it thinking, okay, I've got this great biology here. I'm going to do one proteomic experiment that's going to answer all 4 of these questions at the same time. But what'll happen is that you'll go, now that I have this data and I'm looking at it, I realize I should have done this and this and this to do another proteomic experiment. keeps us in going here. Okay, I want to talk about, do we need to take a break?